Right, exactly.
So just wanted to make sure we're all on the same page about that. Yeah, so yeah, we can go ahead and get started. So Amy Gladfelter, great to have you giving us a seminar on your recent work. Obviously, you've been in the condensate game as long as just about anybody and have done some pioneering work, and wonderful to have a chance to hear from you.
Great, thanks, Mark. It's really fantastic to be here. I'm just gonna get my
screen. Yep.
PowerPoint
Good.
set up.
Perfect.
Okay, so today I'm really going to be focusing on an area of condensate biology that takes the perspective of RNA, which is a major feature of many condensates, and one of the main things we are thinking about in the condensate world. I often get asked, how did I start thinking about condensates? What got me into this field and this business? And it really came out of a long-standing curiosity and interest in a very special kind of cell, which is a syncytial cell. And so I'll be talking about a couple of different types of syncytia today. First, I'm going to talk about fungal syncytia, which you can see an example of one here growing under the microscope. This is the form that many fungi take, both in the forest when they're growing under a mushroom like this, but also as pathogens in hosts. And what's remarkable is these cells can achieve incredible size by virtue of having many copies of the genome within a common cytoplasm. But fungi are not the only syncytia. I'll be also talking about muscle later in the talk today. But in fact, syncytia are actually found in many different contexts. We see syncytia occurring in a variety of different infections. So here's an example of a plasmodium infection where there's an obligate syncytial stage in the liver in the host. And in fact, many viruses also form syncytia as part of the progression through an infection, including SARS-CoV-2 is known to be able to form syncytia and uses this to potentially transmit between cells. A number of tumors also become syncytial. Here's an example, just a diagram of a glioblastoma tumor, where becoming syncytial is associated with tumors really becoming more stress resistant and also resistant to different therapeutics. And the idea here is essentially just like that fungus in the forest, where a very large cell can make long range communication over large areas and integrate information over a lot of different areas. The same might be the case for tumor cells that achieve large sizes and can cover a lot more territory when they become syncytial. And then finally, I won't talk much about this today, but we have work in the lab looking at the syncytial trophoblast of the placenta. And that's this dark blue cell in this diagram here. And this is an amazing cell in that it's performing functions as diverse as sort of different tissues can even perform, but it's actually happening within a common cytoplasm. And it's really this general feature of syncytia that's fascinated me for many years and drove me into this field. I was really led to this field by trying to understand how these very special large cells that contain many nuclei can perform such distinct functions at once. So in the case of the placenta, the syncytial trophoblast cell, you're looking at endocrine function, excretion function, diverse metabolic functions, immunity, all within a single cell. And it was really in trying to understand how you could get compartmentalized function within a continuous cytosol. That's what's led me to here today. And so what we found in my group is that it is through the formation of condensed droplets, particularly of RNA and protein, that you can create functional neighborhoods or territories within a syncytium that allows for this regionalized activity. And so what we think is sort of special about condensates in the context of these large cells is it allows these cells to exist sort of in these two states. One is this highly functionalized state where there's discrete areas that are doing different things. But because of the nature of condensates, because they're dynamic, they can assemble, they can disassemble, it allows these cells to also exist as sort of supercells where they're very large, and actually maybe everyone is acting in unison in those cases. And that this ability to sort of transform between these more unicellular and more multicellular identities might be a real functional advantage of being in a syncytium and also exploiting condensates. So how do condensates form? What I think is one of the most interesting features of this field is thinking about how these sort of nanometer scale interactions between the constituents that condense RNA binding proteins is what I'll be focusing on today, but obviously proteins that in general have some sort of disordered or low complexity sequences, but many of them also have the capacity to interact with nucleic acids, such as RNA. And RNA generally will have domains within it or features within it that allow it to interact with RNA binding proteins. And so our current understanding of this is that it's essentially a series of weak multivalent interactions that can drive these molecules together at the nanometer scale. But the many of these molecules interacting together, of course, will nucleate and ultimately drive the demixing process, leading to the formation of a condensate. And obviously what we detect are these mesoscale droplets. And so what's really interesting to me is thinking about how the strength and the spacing and the information in these nanometer scale interactions ultimately inform the mesoscale physical properties and ultimately function of the condensates. And so it really is the placement and the strength of the interactions at this small scale that are going to determine features such as viscosity, surface tension, the ability to fuse, the size of the droplets, the sphericity of the droplets, all of these things that we can actually measure and that we think are probably important for function. And what I'll really want to be focusing on today is thinking about the RNA component of this. And so there's been a tremendous amount of interest and focus on the protein component, but what about the RNA? So RNA is, everyone knows, is a very common constituent of many condensates in the cytoplasm and also in the nucleus. And so what I think makes it especially powerful for condensate formation is sort of the variety of different features that it has. So one feature that I think has been most thought about the most in the field is that it has a uniform negative charge. And so obviously this charge is going to be, the amount of charge is dependent on the length. But I think this is really only the beginning. And so there's a tremendous amount of more information within an RNA molecule other than just charge. So multivalency is a feature of many specific RNA protein interactions where a given RNA will actually have multiple, in general, binding sites for an RNA binding protein. And these are generally low affinity interactions. And so having multiple ones helps facilitate that interaction with the protein. We know that RNAs can be highly variable in size, just to the different sizes they're encoded, but also through alternative splicing, which I'll be talking about in the later part of my talk. But one thing I think that's potentially not widely appreciated is how large RNA is. RNA molecules can be on the order of 100 times as large as a protein. And so this is really important for thinking about their potential roles as scaffolds and also thinking about their mobility and the idea that they may be actually quite long-lived constituents of condensates. So we've thought a lot about how the structure of RNA can influence condensate specificity. I'll talk about that published work in a few minutes. And I'm just showing you here some pieces of RNAs that we've studied. And what's remarkable is that RNA can take on sequence encoded structures like this. There's a whole ensemble of different sorts of structures that any given sequence can take on. And then these secondary structures obviously give rise to tertiary and then ultimately even quaternary structures that are sequence encoded. And then finally, there are specific mechanical properties. There's specific flexibilities depending on the degree of disorder within an RNA sequence and order within an RNA sequence. So properties that we think about often for, say, features of the cytoskeleton, such as persistence length or sort of the stiffness of the polymer, I think can ultimately become really relevant for thinking about what RNA may be contributing to condensate properties. So I'll talk today really about sort of some published work that we've done on this fundamental question of what is the basis of molecular identity, and how these features of RNA are critical, we think, for specifying the composition of a condensate. We spend a lot of time talking about or thinking about sort of cell biological context of condensates. How are they scaled? How is their position controlled? Won't be talking about that so much today. And instead, I want to spend a fair amount of time dwelling on the question of how RNA may contribute to the material state of condensates. But to begin with, let's think about the molecular identity. So I'll very quickly introduce you to some of the molecules that we think about in a model fungal system. So this is a filamentous fungus that is a syncytium. It's the problems that I was fascinated with in this system are shown here in this video, where one feature is the nuclei divide asynchronously, even though they're all bathed in the common cytoplasm. This suggested to me that there was some sort of compartmentalization or organization of the cytosol. This was when I was a postdoc, and so it was many years actually before anyone had thought about a condensate, but I was imagining sort of a general insulator that kept cell cycle components from freely diffusing. The other feature in this cell that's so interesting is the way it grows. It grows by this branching morphogenesis. So all growth is directed at tips. This is a feature, it's very similar to how neurons develop and neurons grow. And I think a lot of the problems that syncytia face by being very large and having to compartmentalize their cytosol are also faced in large neuronal cells. So for each of these branches to come out, basically the cytosol has to be regionalized or specified in some way, again, suggesting there might be organization in the cytosol to break symmetry for growth. So the molecules that we now know that are important for both the cell cycle and also cell polarity is first a protein called We3. This is a polyglutamine track containing RNA binding protein that condenses into liquid-like condensates at sites of polarized growth and in the periphery of nuclei, which are sort of black holes in this movie here. So the polyglutamine track at the protein is essential for these droplets to form. Here are just some stills of the condensates. And when we delete the polyglutamine track, you no longer see anywhere near the level of condensate formation. And this has major consequences on these two features of the cell. So one is when we eliminate either the protein or the polyglutamine track, the nuclei no longer divide asynchronously. Instead, you see these tracks of synchronous division because the cytoplasm, we think, has become more homogeneous in this area. So the cell cycle is being shared amongst nuclei. And then when we look at cell polarity, we can see the normal cells put out many, many different side branches to create these beautiful mycelial shapes, whereas mutants lacking the ability to form condensates make these very straight hyphae with very few new sites of cell polarity. So we've spent a lot of time trying to understand the mechanisms of phase separation for this protein. And I think one of the most powerful and interesting features of the We3 protein is that it really will not condense with any old RNA. It has real specificity for particular RNAs that are important in nuclear division and cell polarity. So the protein on its own does not undergo demixing under physiological salt and physiological protein concentrations, but it robustly demixes in the presence of cyclin RNA or polarity RNAs. And this has really allowed us to ask some very fundamental questions about how liquid droplets could have distinct molecular identities, because you should imagine, this is the case for all condensates, they should just fuse and mix together into one gigantic droplet, and that doesn't happen. We know that there's a myriad of different condensates of many compositions in the cell. And so in this system, we have one protein, the We3 protein, that can localize different RNAs, and I'll show you that here in the next image. In fact, it localizes, it's important for localizing, these are actually the RNAs that it's regulating. It's important for localizing polarity RNAs, and RNA is important for the cell cycle. And it's forming distinct assemblies with these different RNAs. So it's the same protein component, but different RNA components. And so we are very interested to understand how a single protein could be in these functionally distinct assemblies. And so in published work, we were able to show that, in fact, we can reconstitute this identity, even in vitro, with very minimal components. So if we mixed polarity RNA and cyclin RNA with the We3 protein, we could see that, in fact, they form distinct condensates, even with no other information in the cell. And so this told us that the RNA was really important for this. So we did a simple experiment early on in this study where we melted the cyclin RNA. So we didn't change the sequence at all. We simply melted it, which would just eliminate all of the secondary structure in the RNA. And what we could see is this actually allowed these two RNAs to now co-condense and mix together. And so we went on and did a lot of structural studies to look at sequences in the RNA that we thought might be somehow imparting specificity. And ultimately, after doing a lot of structural analysis, we essentially had a hypothesis that RNAs are able to sort depending on their ability to undergo intermolecular interactions. And so that specific sequences within cyclin and cell cycle RNAs or polarity RNAs were allowing those RNAs to undergo a complementary base pairing and ultimately specify their location within either the same droplet or keep them actually in separate droplets. And we've done a number of experiments to test this hypothesis, both in vitro and also in cells. But here's just an example of one of the approaches. Using the structural studies that we had, we knew what sequences of the cyclin RNA might be able to interact with the foramen RNA if it was available. But the idea is in a normal cell, those RNA sequences are actually tied up in secondary higher order structures. And so we did the following experiment where we melted the RNA. I already showed you this, where they mixed together. But now when we add oligonucleotides to these sequences where they might be able to interact, we can actually block that interaction and restore the segregation, suggesting that there really are RNA sequences that can determine whether RNAs are together or apart. And so what we're thinking now is this is a really important feature of sorting into droplets, is that the secondary structure is essentially licensing which RNAs might be able to interact with one another and which RNAs are not able, don't have an affinity for one another, and they end up segregated into distinct structures. So this work, I think, has implications and lots of applications within the field because I think there's more and more cases where we're seeing RNA-RNA based interactions being relevant for granule formation. So we know from beautiful work from Ruth Lehmann's lab that, in fact, there appears to be high degree of organization of specific RNAs within these germ granules. And this has been postulated to occur through RNA-RNA interactions that sort the RNAs within the condensate. And Ankur Jain has shown beautifully that RNA assemblies, exclusive RNA assemblies can occur from triplet-containing RNAs. And Roy Parker's lab has shown important roles for non-specific RNA self-assembly in seeding stress granules. We think this is probably the tip of the iceberg in terms of thinking about how RNA-RNA interactions are relevant. So there's a tremendous number of questions, I think, on RNA-based specificity in the condensate world. We don't know where in the cell RNA-RNA interactions are initiated. Is this happening in the nucleus after transcription? Is it happening in the cytoplasm? Essentially, how might that be regulated? Are these RNA complexes essentially nucleating condensates? Are they very important seeding events for assembly? Is that a general principle? What is the degree to which specific RNA-RNA interactions are being actively remodeled by helicases in a controlled manner? And Finally, does the composition to different RNAs actually lead to distinct physical properties of droplets? So is the RNA sequence also imparting a physical identity to the droplet? And so this is really the question I'm going to talk about now for a few minutes. So it's well appreciated that condensate physical properties can vary widely. And so these are all examples of RNA-rich granules that vary in terms of their viscosity over orders of magnitude. And in our own work, we've shown that the physical properties of droplets can vary depending on the RNA sequence in a simple reconstitution system, the protein and RNA system I already described. And so here are measurements we did using microreology, where we're just tracking the diffusion, or the mobility, I should say, of microspheres within condensates to estimate the material state of them. And what we could see is that RNAs encoding polarity components are much less viscous than cell cycle encoded components and droplets. And one thing that's really interesting about this is you might think, oh, well, maybe that's just a length dependence. What's really interesting is that the polarity RNA is actually much longer than the cell cycle RNA, and yet it makes actually a less viscous condensate. And so we think it's is something much more sort of interesting and complicated than just simply a length dependence of the condensate properties. And so we're spending a lot of time thinking right now of RNA as a structural scaffold for droplet assembly. And we think that this is a very attractive scaffold for a number of reasons, some of which I'll be able to share with you today. I'm just reminding you here of the great size disparity between proteins and RNAs within the droplets, and the ability of RNAs to really form large systems spanning networks through their ability to have these sort of stable intermolecular base pairing interactions. And so how can we imagine RNA as a scaffold? I think you can imagine sort of two general classes potentially of RNA scaffolds. So one would involve non-specific RNA interactions, and this might be akin to what the Parker lab is postulating for stress granules, where essentially you might have heterogeneous assemblies of many different RNAs, different compositions. Because there's not necessarily sequence encoded specific interactions, these may be more dynamic or more labile. Partly because there's probably a tremendous amount of helicase activity that may inhibit these under normal circumstances, these sort of non-specific interactions. However, it seems quite clear that there's evidence that these sorts of interactions can be induced by stress when translation is paused and sort of RNA sequence becomes available to become tangled. Now, what I'm thinking much more about are sequence encoded RNA interactions. And so what's really special about these sort of interactions and making a scaffold is that you can actually create a mesh of a particular length scale because there's actually sequence encoded interactions that could, in a sense, specify the exact properties. I think this could impart elastic properties. This can also, of course, be tunable and regulatable by helicases. But I think where we're seeing examples of this, it tends to be in developmental regulation, cases where there's a high degree of organization and regulation that is required by the condensate. So here's some example of work we're doing to look at the importance of RNA-RNA interactions in the material properties and sort of scaffolding functions. So based on some structural work and predictions that we've done, we find that there's a large area of predicted interaction just between two molecules of the cyclin, CLIN3. This is one of the cell cycle components I was talking about earlier. So there's a large degree of potential homologous interaction here. And we've generated mutants in this that substantially disrupt the pairing and we think substantially change the higher order structure of even just this pairwise interaction. And what's really remarkable is these sequences are largely similar. There's just a very small handful of substitutions. But when we look at condensates made from either wild type or these pairing mutants, we see a dramatic effect on droplet size, the assembly rates, They ripen much slower. We see very different fusion rates and really different rates of hardening. And so this is based on, relatively small changes in the sequence that are having big effects on the overall droplet properties. So that's sort of an example of thinking about RNA-RNA interactions. I think another key piece of RNA acting as a scaffold and potentially being set up as a scaffold are protein-RNA interactions. And so this is where we start thinking about the valence of the RNA and the number of binding sites that it has for a protein within a condensate. And here too, we've seen that small differences have a big effect. So if we look at an RNA that has 4 potential binding sites for the We3 protein, we get condensates forming. If we add a single other binding site, so that we have now 5 binding sites, we see a dramatic change in the size of the droplets that form. And I should say this is the sort of native number of sequences for the structure. So taking one away, and I should say this is also relevant, we think that it's positioned at a distinct location from these other sites. And so it's not just having a certain number of binding sites, but we think it's also the position of these binding sites that's really relevant for, again, determining the material state of the condensates. So we have a large battery of wild fungi that we study that have varying different sequences, and we've collected these fungi from many different climates. And this is part of a study that we haven't published yet, but we're really excited about because we think we're able to look at essentially adaptive sequence changes that are really relevant for allowing the fungus to adapt to growth at different temperatures. And one of the things that's so interesting is across our collection of wild isolates that we've collected from a lot of different climates, what we're seeing is that the targets of We3 actually increase in their valence as the temperature, the ambient or sort of average temperature in a climate drops. So if we see sequences that are from more northern climates, they tend to have many more We3 binding sites. So the valence is increasing as a function of temperature. So we think the valence in the RNA is actually probably a really critical additional component here. And one of the things that's interesting that we're trying to do now is start trying to look at the intersection between RNA-RNA interactions and valence. And so one of the sort of interesting features of these wild isolates is this warm weather isolate. These are cyclin sequences from a isolate from Florida. It has this ability to undergo pairing here, and we think this is actually helping it to form larger and larger condensates at higher temperatures. Whereas if we take an isolate from Wisconsin, in fact, these RNAs appear to be disrupted in this pairing area. There's quite a bit of variation within the sequence in this area, and this is actually changing the droplets' ability to form at higher temperatures. And so what we're imagining is that maybe there's increased valency in these colder climates that's sort of compensating for the lack of intermolecular pairing that you see in the warmer climates, and that's missing in these colder climates. And so the point I want you to take from this is that I think there's a tremendous capacity for variation within the RNA sequence, and that there's essentially a balance between valency and RNA interactions that's able to give you essentially different strengths of interaction that may be tunable in terms of both the material properties and also the environmental responsiveness of the condensates. And so a lot of our work is really trying to imagine and come up with ways to actually measure the long-range RNA interactions that we think are important for giving RNA this potential scaffolding property for droplet assembly. And so what I'd really like to propose is there's been a lot of emphasis in thinking about proteins as clients and scaffolds and condensates, but I think we really need to think about RNA similarly in these classes, and that RNA may be playing a critical structural scaffolding role, and there may be other RNAs that are clients within condensates. And in the last few minutes here, I'm just going to talk a little bit about, I think, a really important feature of RNA that's also probably been a little bit underappreciated in the condensate world, and that is the potential of alternative splicing that occurs throughout higher eukaryotes that can alter essentially all the features of RNA. It can influence the ultimate charge if it's in being driven through a complex coacervation by affecting the length, the number of binding sites, depending on which exons are included. Obviously, it can affect the size and the structure. All of these properties that are sequence encoded are going to be influenced by alternative splicing. And I think everybody appreciates this, but there is a tremendous potential for variation here within a single gene to essentially create both high degrees of variation at the RNA sequence level, but also influence ultimately the encoded protein, which is what's so often thought about. But I think it's really important to think about both of these features in the context of condensate biology, because it's appreciated that many RNA binding proteins bind to their own RNA. And so that alternative splicing can have potentially very potent effects on ultimate condensate properties by influencing both the RNA species and the protein species that might be present. And so I'm not, talking here about the condensates involved in splicing. That's sort of a problem for another day and probably another speaker. But instead, what I'm talking about here is sort of the effects of alternative splicing. And so what's got us thinking about alternative splicing as an important problem is sort of our broader interest, again, in syncytial cells. And so as I've already talked a lot about fungal cells, skeletal muscle cells are another really important multinucleated cell where it's known that nuclei can essentially be autonomous or independent from one another, and particularly nuclei that are in the vicinity of neuromuscular junctions express different RNAs, and that in the process of myotube formation, that there's tremendous amount of nuclear to nuclear heterogeneity. And so this smells a lot like the nuclear asynchrony that we saw in fungi. And so this got us starting to think about myotube formation and whether there might be spatially distinct cytosolic territories in these multinucleated cells. And what drew our attention to splicing was this protein called FXR1, or Fragile X-related protein 1. FXR1 is a large gene, and it's highly expressed in muscle, in myoblasts and also in myotubes. So the way myotubes form is through myoblasts differentiating, changing shape, and then fusing to create multinucleated myotubes, which will then ultimately go on and give rise to muscle tissue. It's important to note that this happens in development, but it's also happening in repair and is really relevant for muscle regeneration as well. So FXR1 undergoes a major shift in its isoforms that are present between the myoblast state and the myotube state. And so specifically, myoblasts are characterized by these shorter isoforms, the A and B isoforms, whereas during the process of differentiation, we see an inclusion of these two additional exons, 15 and 16, in the myotube form. So what's important about 15 and 16, which hopefully everybody can appreciate, is that FXR1 contains a very large intrinsically disordered domain at the C terminus. It has an RGG motif that we think is relevant for RNA binding, as well as two additional RNA binding motifs, KH1 and KH2. So the inclusion of 15 and 16 alters the sequence and the length of the C-terminal disordered sequence. So because of this developmentally controlled splicing that changes the disordered sequence, we were very interested in whether this could be a protein that's important for patterning and sort of setting up territories of cytoplasm within the myotube. While we were studying this, we were really emboldened and excited to hear about a study, a genetic study on FXR1, identifying recessive mutations in a muscle-specific isoform of FXR1, essentially a variety of different mutations that either frame shift or eliminate splicing of 15 and inclusion of 15 and 16. So essentially leads to a truncated or altered form of the protein so that you don't get that sort of extended differentiated form of FXR1. So this is a recessive disease. And what was really interesting in this literature, and these are in mature muscle cells, you can actually see organized assemblies of FXR1 in the skeletal muscle. In patient mutations, you interestingly see a disorganization of this. You also actually see things that sure scream to be condensates, the sort of larger droplets that form. You see a much more heterogeneous size distribution compared to the wild type, or in some cases, a diminishment of condensates. So importantly here, we see splicing associated with the disruption or the alteration of an IDR. So this is work that we recently published and as in the JCB if you want to read more about it. But essentially what we were able to show is that in myoblasts, here's just a scheme looking at the disorder tendency so that you can appreciate the myoblast form has a much shorter intrinsically disordered sequence compared to the myotube isoform here, which is this extended disordered sequence. And this is endogenous protein, how it's localizing within cultured C2, C12 cells. You can see they're syncytial. These are very early on in their differentiation, so they have a mixture of these isoforms. But you can start to see that, in fact, there are condensates that form, and we're able to show that they confuse and they appear to be dynamic. And this is just a pelleting assay showing essentially that before differentiation, the protein is mostly soluble, but as the cells differentiate, you can see more and more of it assembling in the pellet, consistent with some sort of higher order assembly and maybe a more solid or gel-like core. And we did a number of different experiments with this protein. This is just an example of trying to understand essentially sort of how FXR1 behaves. And so we made a number of different mutants in RNA binding domains. And what was very clear to us is that the FXR1 protein requires RNA binding in order to undergo phase separation. So here's a mutant that is lacking both the RGG and the 2KH domains, and you can see that the protein is no longer visible in puncta of any sort and appears to be in this sort of soluble state. But one of the things that was most surprising to us from this is when we looked at this last variant here, which is a shortened form of it, and it's essentially very close to the A isoform, so the myoblast form of it. So it's missing a large chunk of the IDD. And so recall that, in fact, we hypothesized that maybe the IDR was getting longer as a function of splicing in order to promote phase separation. But it turns out in shortening this, we actually got even larger condensates and more condensates at lower concentrations of the protein, suggesting that this was really not such a simple story as just a longer IDR is more and better for phase separation. But this is a much more complex relationship between the sequence and the condensate state. And so we're still really trying to understand this at this point, but we know that FXR1 can bind to its own transcript and that this facilitates phase separation. And how we're kind of imagining this right now is that alternative splicing of FXR1 is likely relevant both at the level of the protein and at the level of the RNA. And so here in myoblasts, we have a smaller IDR, smaller FXR1 RNA. And this is actually associated with more liquid-like assemblies, we think, early on in development. But now as we differentiate more into myotubes, the IDR is expanded. This also actually increases phosphorylation sites in the IDR, and I didn't have a chance to talk about that. We have a little bit of this in the paper. This also expands the length of the FXR1 RNA, and that these two features are actually associated with more gel-like or solid-like assemblies. And so the longer IDR and RNA are actually associated with less dynamic assemblies of FXR1. And so, we don't fully understand the implication of this. We think that this is regulating the translation of FXR1, but that's really future work to be done. But I think the lesson in this is simple predictions just based on sequence are really very difficult to necessarily truly predict well. So I'll just leave you with this key big, or I don't know that it's a big idea, but this key idea that RNA we think is a really critical structural component for droplet assembly, and that the sequence of RNA has really important properties, both to specify which RNAs end up in a condensate, but ultimately the network of RNAs that can form that is important for these larger mesoscale physical properties that I think we all envision play an important functional role in condensate biology. So I just want to thank my lab for making this just a phenomenal job and being so excited to come to work when we're allowed to come to work. And this work was primarily done by a current postdoc in the lab, Ben Stormo, and Christine Rodin. The lab is made up of people who are really span the spectrum of biologists to biophysicists to actually theoreticians. And so it's been a really privilege to train all of these young scientists. And I also want to thank my great collaborators and my funding sources. And thank you for giving me a chance to share these ideas.
Thanks, Amy. It's great, wonderful stuff. I'm sure we have a lot of questions, but I'll ask one while people are gathering. It's first of all, it's great for somebody like me as an old protein guy to hear a talk like yours, which is just trying to get us all to think about the role of RNA differently. Mm-hmm. And so it's interesting when you rattle off all that wonderful slide you showed a couple of times, all the different factors that matter, charge, multivalency, the size of the RNA, structure, right, the dynamic nature of it, all of that. And so each of those is in itself complicated to study.
Yeah, exactly.
Yeah, and so it's interesting to think about in any given situation, which of those factors will be the most relevant, or is it always, probably is always some mix of all of them,
Yeah, I mean, in our hands, we often see mixtures, right? But you're right, it's hard to disentangle all these. And I think this is true, honestly, for protein biology also in in the condensate world, right? If you shorten it, you're eliminating sequence, but you're also potentially changing other physiochemical properties, right? So for sure, small sequence changes can change the structure. They potentially are changing the valence. We've seen major effects on length. Really, I think length is a really important feature that is critical. I think a lot of times what one is going to need to do for studying RNA is to move between simplified models of the RNAs of interest, right? So sort of toy molecules. Mm-hmm. And I think they're dangerous in some ways because they're simple and they're not necessarily, have the complexity, but so that you can try to isolate one or two of these features at a time and then move it back into the more complicated molecule that, you can actually experimentally probe with a little bit more knowledge and simplicity.
Mm-hmm. Yep, exactly. Anne, did you have a question?
Yes. Amy, I know your talk. This is your talk was about RNA, and but I'm curious about something you mentioned in the beginning in your.
Oh, I just lost you.
I'm sorry. Can you hear me now?
I can hear you now. Yes.
Yep.
Sorry. I'm a virologist by training, and you mentioned viruses that cause syncytia, right? And like in herpes simplex virus, there's these mutants that cause syncytia, and they're glycoproteins. And it really seems to be driven by, there's several different glycoproteins that can cause syncytia. Are there any rules or basic principles that can be generalized from these single glycoproteins in the viral systems that seem to cause syncytia and allow the fusion of these, large syncytia fusion across, the entire cell, the dishes in a cell?
Yeah.
Compared
Yeah,
to these amazing organisms that you're describing that,
Well, I think there's a lot of ways to think about the problem you're addressing. I think one is the mechanism of fusion and how you become a syncytium. And whether those glycoproteins, there could actually be sort of some phase separation mediated processes that are helping promote fusion actually, right? And I think that's possible, although I don't know of anyone who's shown that at this point.
Yeah, I haven't seen that.
But we do have a project in the lab actually looking at sort of how membranes may facilitate protein-driven phase separation, where there maybe not be as much RNA present in those sorts of membrane fusion events, and so these could be sort of protein-driven, and I think glycosylation is certainly on the table as a very important post-translational modification that I think will be really relevant for phase separated condensates. But I don't think anybody's shown that for fusion, cell fusion at this point, but it wouldn't surprise me. And then the second part is sort of once you're a syncytium and there's this massive state, sort of what is that bringing these cells functionally? For a virus, it really may be a means to very stealthily expand, right?
Right, you're not exposed to the external environment.
You're not exposed, right? But I think, there could also be other advantages to this in terms of just how viral replication is controlled spatially. And it's something that we've really not studied ourselves at this point. But I've been thinking more and more about in light of SARS-CoV-2, actually.
Mm-hmm. Yeah. And it's interesting because some viruses can often do both. They can do both. They can spread by fusion in some situations, and then spread by lytic infection. And there's obviously advantages to go both one way or the other.
One way or the other. Yeah, absolutely. Absolutely. Yeah.
Okay. Thanks. That's interesting.
Yeah. Thanks.
That was a great talk, Amy. Really enjoyed it.
Oh,
as
thanks.
usual.
Hi.
Hi. So, you talked about helicases and how by changing the structure, they modulate the valency and so on. I was wondering if you can comment a little bit about post-transcriptional modifications and how those contribute to real time.
Yeah, so, I think this is something I should have put on that slide that I neglected to put on all the wonderful things of RNA. It should be there, right? So post-transcriptional modifications are for sure probably a major modulator that the reason it wasn't on my slide is because it's basically M6A is one of the main ones. This has been, linked to condensate biology, right? And I think it's just because, it's a bit of a new world of mapping all the post-transcriptional modifications that are on RNAs. And so it's just a few steps behind sort of thinking about this in terms of protein modifications. But I think we should envision post-transcriptional modifications exactly the way we're seeing effects on translational modifications on proteins, right? This is a great way to modify shape charge, conformations, binding proteins, like it's essentially critical. So it was an absence on my slide of all the wonderful things. I didn't have room for all the amazing things that RNA can contribute to a condensate. So yeah, that's again, very early days, but I mean, I think M6A is the poster child for now, where that's actually probably a modification happening within a phase separated assembly. I think that is very, very early days, and partly it's hard. It's hard to make in vitro, modified RNAs where you have site-specific modification, for example, right? Like these are really hard experiments right now to nail really what a specific modification may do at a specific residue, right? But it's something we're just beginning. We're actually sort of starting to try to map post-transcriptional modifications that are on the model fungal RNAs that we're studying, because we think in a fungal system this will be simpler. There'll certainly be less different kinds of modifications, so that the code should be a little less complex. So I'm hoping in the future that we have more insight into that, but that's a hard problem.
So I think you probably said it and I may have missed it. Do the cycling in polarity RNAs ever colocalize in vivo?
Yeah, we haven't found them, but we think they must actually, because there is a capacity for them to mix. And so we haven't exhaustively looked under different environmental conditions, but my hunch is that there probably is some either developmental context or stress-induced context where it would make sense for them to actually co-assemble. And there actually may be a context of that. And whether that promotes their translation or maybe inhibits their translation, I'm not sure, of course, at this point. But I do think there's a reason there's some capacity there, because there are fairly reasonable tracks of homology that can enable them to interact if they're available. So, potentially a stress-induced helicase or something could potentially expose that and allow them to then co-condense and coordinate maybe better nuclear division with growth.
Great, thank you.
Yeah,
Hi.
go ahead, please.
Sorry, Mark.
Please, go ahead, Avi.
Hi, Amy. Very nice talk. Really exciting, the FXR1 story, right? So I just was wondering about the things that you presented towards the end of the story, right? That the changing material properties associated with the development of myoblasts to myotubes. So have you also looked at this phenomena in kind of, in dynamic living myotubes? And, if I remember the cartoon that you showed, it seemed like this condensates kind of assemble around the nucleus. Could that be a way of mechanism that, they make sure that there is a isolation or segregation in the cytoplasm, and the property actually does help them in being isolated, or how they move around once
Yeah, so we really haven't pushed the imaging to these later stages of development, mostly because it's really hard to image them at that point for us, because we're not muscle specialists. We're not using primary cells, and they start to peel off and they become really difficult to work with. And so unfortunately we didn't really in the end nail that model that this matters for nuclear functionalization. So that remains a possibility, and at this point sadly I don't have evidence for that yet.
Okay.
Other questions? Yeah, one thing about helicases that's interesting is that unfortunately there are not a lot of good tool compounds yet. And that makes it a little harder to try to tease apart the role that they play. Of course, there are other, you could use CRISPR and other knockdown methods instead, but from a pharmacology standpoint, there's not too much available yet.
Why do you think that is, Mark? Is it because just people haven't looked, or is it because there's something about their structure? Like, have you looked at
Yeah, they're difficult targets. People have tried actually quite a bit. There's a lot of examples in the literature, and they often end up being quite challenging to make good, small, simple, selective drug-like molecules. I think there'll be, I think there's going to be sort of a new wave of people trying to go after helicases again. I'm seeing more evidence of that in the literature. So hopefully we'll, in this next round of research efforts, we'll have some better tools.
Right. Okay.
Yeah, and it's clearly a very important area. So
yeah.
that's good. Any other questions? Well, if not, Amy, thanks again. Wonderful lecture as always. Thanks for sharing your thoughts with us.
My pleasure. Thanks for listening.
Oh, it's great stuff.
Yeah, thank you so much. That was great.
Yeah.
Thank
Take
you.
care.
Thanks so
Okay.
much.
Bye.
Take care. See you.
Bye.
Bye-bye.